Okay, we're starting chapter 4, so there's a new Moodle quiz open, and I'm going to introduce some ideas about cells, about cell theory, cell size, why cells are important, and in particularly, I'll start describing the structure of prokaryotic cells. Um, and so I got a little far side here on early microscopes, and when probably the magnification was lower, but the resolution was probably pretty good. And biologists really didn't understand until microscopes that most living organisms were really, really small. Um, they knew, okay, mice are more abundant than people. Mosquitoes might be more abundant than mice, but they really didn't understand that it was the uh, abundance of bacteria and other organisms that were single cells weighing much less than a gram, that those were the most abundant. And we didn't understand that until microscopes. So Robert Hooke, shown in the lower right there, and Anton von Leeuwenhoek, shown in kind of in the middle on the left. These were the two early guys that really uh, popularized microscopes as scientific tools, as tools for seeing the invisible, and that might be important. And not only did they observe things, but they recorded what they observed and shared that, which is important in science. Here's a von Leeuwenhoek's microscope. It had a series of screws to precisely position what he was looking at right in front of a lens. And with this, he was able to see bacteria, extremely small cells. And so he had it looks primitive, but it was actually a fairly high quality microscope. Um, and so here we see, okay, with the eye, you can see things to the size of maybe human eggs or frog eggs. You can't really see much detail. But with the light microscope, you can get down to much finer details. And then we can even use electron microscopes to see much smaller things, even large molecules and viruses. So when we talk about cells, we're often talking about micrometers. And so 1 micrometer, which is a kind of that funny looking that equals 1,000 millimeters, which are MMs, or uh, one, excuse me, yeah, 10 to the minus sixth, or 1/1 millionth of a meter, or 1/1000th of a millimeter. Okay, and so most bacteria are measured in micrometers, and most are less than 5 micrometers in length. Sometimes we'll measure things in nanometers. Those are NMs. 1,000 nanometers equals 1 micrometer. So a virus is often measured in nanometers. And there's E. coli. E. coli has a width of less than 1 micrometer and a length of maybe about 2 or 3. So very hard to see with the light microscope, and impossible without it. Most of the stuff we're going to look at is made from drawings or pictures of light microscopes or electron microscopes. Light microscopes, because of the wavelength of light, they can see structures that approach about 200 nanometers in length in ideal conditions. So a little bit less than 1 micron. Okay, that's really ideal conditions. Really, like for our microscopes, we can see things about 1 micron. So not ideal conditions. Electron microscopes are maybe 1,000 times more powerful, so they can magnify things maybe 100,000 to 1,000,000 times. So they can resolve structures down to, well, maybe less than a nanometer. Okay, and so here we have a picture of a light microscope on the left and an electron microscope on the right. And here are pictures taken with each of these. So much finer resolution. Light microscopes are just a series of magnets that catch light that's coming up through a specimen. And usually the specimen looks dark against a bright background. If you want it to look darker, you can stain it. And you can do various things with the light. You can change the wavelength of the light, use blue light or something like that. Electrons have a much shorter wavelength than light, so they travel in a straighter line. And here you don't use a glass lens to focus light. What you use are magnets to focus beams of electrons. And so you can get magnifications up to 100,000, and the resolution is really what's spectacular. Like this fly, the magnification's not that great, but the resolution is just terrific. You could never see this kind of deep detail with a light microscope. Okay, so we'll use two types of electron microscope pictures. One is a scanning electron microscope that takes a picture of the surface. And so the beams hit the specimen and bounce off, and that's when the collector sees it. And so you get a picture that looks like this. It's a picture of the surface of that. And again, really nice resolution, much better than a light microscope. Magnification here is generally less than 20,000, anywhere from a couple dozen to maybe up to 1,000. The other type of electron microscope shines electrons right through a sectioned specimen. And again, this has to be stained, but here we can get to these really high resolutions. And so really good for seeing stuff. So when I talk about cell features, I'm kind of saying, here's what we saw with the light microscope and the electron microscope. All right, so cell theory, this is a really, really crucial thing. All organisms are made of one or more cells. All life processes occur within cells. Smallest cells are the smallest things that are truly alive. They do all the things that a living organism does. They take in energy, they get rid of wastes, they respond to their environment, they grow, they reproduce, things like that. And cells come from pre-existing cells, so cells are capable of reproducing. Okay, one of the key, why are cells so small then? Well, cell size is limited because all cells rely on diffusion. Okay, diffusion is the random movement of molecules from areas of high concentration to low concentration. Diffusion is needed for organisms to get energy, to get rid of waste products. They rely on osmosis for movement of water. Cells are limited because these processes are only effective when the distance is relatively short. If a cell is really big, it has to move things across a long distance. Okay, and so here is this on this little chart on the bottom. As the cell radius gets larger, its surface area increases, but not as fast as the volume increases. And you can see that here. And so you wind up, the center of the cell winds up getting rid of its waste products and it can't get nutrients. And so to make up for that, we have small cells. Okay, these are fundamental ideas to biology. Cells are small, cells are alive. And biology has changed a lot since I was in college, and that was the previous millennium. But there are some core ideas that are the same, and cell theory is one of them. Cell theory is old. It developed over about 100-year period in the 1800s, but it's well documented. We don't find living things that aren't cells. Sometimes cells can do funny things. They can fuse. Sometimes they get big and flat, but everything is a cell, and cells generally kind of work the same. They have DNA, they have RNA, they make proteins. Okay, so biologists identified all living organisms contain cells, and then they've kind of divided the world up into two basic cell types. All living organisms either have prokaryotic cells or eukaryotic cells. Prokaryotic cells are small. They're very simple. They have no internal compartments. There's just one fluid-filled chamber inside the cell. They have no little organelles that are doing separate things. These are bacteria and archaea, and they appeared a long time ago in the fossil record. And these are the most diverse type of cells, at least chemically. They can do chemical reactions that the other type of cells can never do. The other type of cells are eukaryotic. They are much larger, by often a factor of 10. They have lots of little internal compartments, including always a nucleus, but lots of these little membranous organelles where they do special things. In the history of the Earth, eukaryotic cells are new. Okay, they haven't been around half as long as prokaryotic cells, but these are all the things we can see. Truly visible organisms, multicellular organisms, are all eukaryotic cells. All right, so let's talk about prokaryotic cells then. These are nice in that they're simple. They have one fluid-filled compartment. Okay, there's no internal division. The compartment contains a region called the nucleoid that has one DNA molecule, but that region is not set off by any membrane. All these cells have a cell wall. They all have a cell membrane. Many of them have a flagella that rotates. And they all are doing kind of the same thing. They're making enzymes and using energy. These types of cells fall into two domains. The archaea, which includes some unusual anaerobic organisms, and then the bacteria, which are more familiar. Bacteria include things like streptococcus, staphylococcus, tuberculosis, lots of disease organisms. Those are all there. They, in many ways, they do many things the same. Okay, they even though there's two domains, when you see a flagella in a prokaryotic cell, it's always the same. It has a hook. It's made up of a single protein. It passes through the membrane and the cell wall, and it rotates. These are they're little motors, and they use hydrogen ion concentration gradients to cause a rotary motion, and it kind of works like a propeller. And so you see this same kind of flagella. It suggests the common ancestor of all prokaryotes was a single cell that had a bunch of features: a cell wall, a cell membrane, and this flagella. Prokaryotic cells are found in many environments. Okay, aerobic, anaerobic, groundwater, bottom of lakes, mud, the surface of our skin, inside the human body even. In fact, how many cells, prokaryotic cells, are found in the human body? Billions, trillions, 10 trillion, 100 trillion, a million trillion, or 1,000 trillion? Well, the answer depends on where you go, but the probable, the old guess used to be 10 trillion, but now the guess is probably about 40 trillion. And that prokaryotic cells aren't everywhere. They tend to be on our skin and in our large intestine in large numbers. But wherever they are, they're abundant, more abundant than eukaryotic cells. Okay, I should mention a little bit, most of those are going to be bacteria that are on our body and in our body, because bacteria can tolerate oxygen. There is a group of prokaryotes called the archaea that can't tolerate oxygen, and they show some more similarities to our cells, but they are prokaryotic. Okay, so we'll stop there. We'll review this on Monday, and we'll start maybe talking about eukaryotic cells.